Hello everybody, my name is Michael Crawford. I'm the Chief Executive Officer of the Soil CRC, the Cooperative Research Centre for High Performance Soils. And it's my privilege and honour to host today's webinar, the last in the series for 2021 as we approach Christmas. I'd like to begin by acknowledging the traditional owners of the land upon which we meet today, and I pay my respects to elders past and present. Today's webinar has attracted one of the largest list of registrations for the year in our webinar series, which is probably no surprise given the interest in soil carbon and the interest in regenerative agriculture. And when you put the two terms together in the one title, soil carbon and regenerative agriculture, and they do go very well together, it's an opportunity to be able to talk about the Soil CRC's work in this area to a broad and wide audience. Given that, looking at the registrations, there's quite a few people there that have perhaps been exposed to the Soil CRC for the first time. Just a couple of words about who we are and what we do. So the CRC is funded by the Australian government and by its 40 participants and partners, which includes 8 universities, 4 state government agencies, 8 industry groups and organisations, and importantly, 20 farmer groups or grower groups from across Australia. And it's through those grower groups and the participation of those farmers and farmer representatives in our planning and implementation that we ensure that the research of the Soil CRC is focused on delivering R&D outputs that are relevant to farmers, that help farmers to improve their soil management, give them the tools and knowledge to improve their soil management, and in turn improve their productivity and profitability. The way our webinar works, shortly I'll introduce Gwen, and she'll speak for 20-25 minutes or so. You'll have the opportunity at the end to ask questions, but you can type questions in at any time through the Q&A box, and at the end I'll host that session with Gwen answering the questions as that are posted there, put to the speaker by the audience. You can also make comments in the chat function as it goes along, but put questions in the Q&A box. This webinar is being recorded, and it's been recorded such that we can then post it onto our website and onto our YouTube channel, and you can access all that via soilcrc.com.au. At the same time, I encourage you to connect with us, follow us on Twitter, connect with us on LinkedIn, subscribe to our YouTube channel. You can also see all our past webinars and panel discussions that we've had this year. There's now, and last year too, there's now quite a large list of relevant webinars and panel discussions here. Normally at this time, I'll give a bit of a promo to upcoming webinars and panel discussions. Today, I won't be, because this is the last in the year, and we'll take a hiatus next year, perhaps looking to kick off again in April, May, after a few months. At the start, so give everyone a break from the webinars, but there's plenty on these socials that will keep you engaged and informed about the work that the Soil CRC is doing. So to today's talk on soil carbon and regenerative agriculture. It's been presented by Grelet, Dr. Gwen Grelet, and she's speaking about a project that the Soil CRC has been conducting. And as with all our partners, all our projects, it represents a range of partners, of participants within the Soil CRC, and Gwen will speak more to that point in her opening slides. Gwen works for Landcare Research New Zealand, Manaaki, Whenua Landcare Research, and yes, New Zealand is one of the, Landcare Research is one of our participants in the Soil CRC, and they've been a very valued participant, and it helps to present a different perspective on Australian agriculture, and in turn helps Landcare New Zealand, Landcare Research New Zealand to be informed by what's happening in Australia. Gwen is a senior research in soil ecology at Landcare Research in the land use and ecosystems team. She's based at Lincoln near Canterbury on the South Island. She's a French national, but has been working in New Zealand and resident in New Zealand since 2010. Along the way, she's also worked at the University of Aberdeen and Harvard University in the States. So she brings some quite international perspectives to this area of work. I'm going to leave it there. I'll pass across to Gwen to give us an update on our project on soil carbon and regenerative agriculture. Thank you, Gwen.
Okay, thank you, Michael. It's a really great pleasure to be here. So I will start also with a Māori salutation. So tēnā koutou, tēnā koutou, tēnā koutou katoa. And it's also my way of honoring the kaitaitanga of this land here and the ancestors that are looking after the land here also in New Zealand and also, of course, extended to Australia. So I will. I would like to add a little bit of additional information about where I come from. So as Michael said, I worked in Scotland. I also worked in the US. And currently here in New Zealand, I'm employed by Manaaki Whenua Landcare Research as a soil ecologist. And for the last year, I've also been seconded part-time to a couple of organizations. The first organization is TOHA Fundry. It's an impact investment platform. And my role with that organization is to help inform how the outcome of adopting regenerative practices can be measured and quantified. And my other hat is I'm also seconded part-time, so for both organizations a few hours or days per week to the New Zealand Merino company to do a similar thing, which is to help them set up the science to measure the outcome of any adoption of a regenerative management or regenerative mindset. So that feeds directly back into the research, but I just think it's quite important for the audience to be aware of that as well. So I will start by sharing my screen. Please let me know if this works well. Does this work okay?
Yes, that's good, Gwen.
Yeah, perfect, thank you. So the project that I will talk about, I started just over 2 years ago. It's only a few months from completion. So we are busy looking and scrutinizing all of our data and writing them up. And so I will present where we've got so far and with a greater emphasis on the soil carbon story for that project. So let's see how I share. I stop. Give me 1 second, please.
Yeah, it's all good. We can see it.
Yep, I was just trying to move the slides. Yeah, okay, great. So as Michael said, this is a project that has multiple partners in the spirit of the Soil CRC. So it's a project that we are hosting and leading, but we have as really key partners PIRSE and Whitbelt and RM and Soils for Life. And we're also working with David Davenport, Nicole Masters as a knowledge broker between the regenerative community and the scientific community. Dr. Will Allen help us with facilitating the conversation between these different people who have different knowledge system and different culture, organizational culture and scientific culture. And as we've progressed the project, we've established collaboration and involvement with Dr. Graham Sterling from the Biological Crop Protection, Dr. Paul Mudge and Kate Owen at Landcare Research, and Lucas van Zwitten and Mike Rose at New South Wales DPI. And you'll see some work produced by Ryan and James, who's a talented graphic designer to help us with our communications. So I first provide a very quick introduction, and this is the $1 million question. What is regenerative agriculture? There's a lot of statements and definition, and there's also a lot of resistance in the regenerative community to actually providing a strict definition for many reasons. So what I've done is I've pulled up from 2 key websites in Australia, and we've actually published a couple of reports recently in New Zealand discussing this topic. So I've tried to bring the Australian perspective. And so from the Regenerative Australian Farmers website, regenerative agricultural practices build soil carbon, increase water retention, support soil biology and ecosystem biodiversity. From the Department of Primary Industry and Regional Development in Western Australian, what I like this is bringing the word principle, because a lot of people in the community, the regenerative community, are trying to shift the emphasis away from practice to principles. And so the principle of regenerative agriculture and regenerative pastoralism is to enhance natural ecosystem services, resulting in sustainable production, improved natural resource base, healthy nutrient cycling, increased biodiversity and resilience to change. So the overall message is we're trying to shift the way we are managing the agro-ecosystem so it delivers a resilient system, and it enhances biodiversity, whilst still being able to keep in business and keeping production going. We thought that it was really important to take a co-innovative approach in designing our experimental approach. So I'll tell you just a few words about that. So this is the reason why we worked with Dr. Whenua. That's his specialty, is helping people from different cultural or knowledge background to work together. And the idea is that because we sought to actually roll out the science about regenerative agriculture, we needed to include the perspective that leading regenerative farmers are adding and the way they are seeing their system, which might then impact on the way we should conduct the science. So what we did is at the start of the project, through the network of our Australian partners, we invited to come on board 10 leading farmers that have a huge reputation and a role of leadership in their community. And so most of these farmers came with a regenerative hat, but not all of them. So we also including the perspective of conventional farmers. And what we did is we asked everyone, so everyone in the team that I've presented and the farmers, so all the people that were consulted were, half of them had a practitioner background, and then others were scientists and others were, say, project managers, knowledge brokers, facilitators. So there was a range of perspective in the people that answered and contributed to that small social study. And we asked them from right to left 3 questions. If you had the choice of designing a research project, what research question do you think we would need to ask to understand the role of soil carbon in regenerative farming system? Middle question was, what question do you think we would need to ask to design research to better understand how regenerative farming system work? And the final question on the left was designed to ask, what are the research questions that would need to be asked to understand the barriers or the levers to adoption of regenerative agriculture? So that's targeting more the broader agri-food system. We took all the answers that everybody gave us and we adopted a theme analysis, which is a standard social methodology. We did that at very high level. It's a very small data set that we have. And we basically identified 9 broad themes. And so these themes cut across all 3 questions that we asked. So all 3 levels of looking at the system, the agri-food system, the farm system, and the soil carbon. I will tell you just a little bit more about this in a minute. We use this to inform the setup of our experimental design. So the first thing we did is we took these themes and we looked at how these could be converted in things that we would like to measure. So from top to bottom, for example, for one of the themes was biophysical properties, and there was a huge emphasis on better understanding biophysical properties of regenerative system. One of the things we did is we set up an experimental design that increased the density of data acquisition from regenerative system compared to other systems to which we might be comparing the regenerative system. There was question about biogeochemical cycle. So we measured enzymatic activity and different type of soil carbon. Biology, there was a lot of emphasis on understanding better biology mediated processes. So we looked at nematode community and enzymatic activity, and the list goes on. And so there were two themes that we couldn't address because of the scope of the work that we were tasked to do by the Soil CRC, and because of the length of time of the project and the funding that was available. This was a project that was set up as a pilot project to optimize the way we should conduct research for better delivery and much more efficient use of resources. Is economic and cost benefit trade-off. We didn't have the resources to do that well. And really understanding mindset in terms of values, purpose, and the decision-making process when regenerative practitioners see the ecosystem in a different way. So that requires a whole lot of resource investment and methodology and expertise that our project wasn't designed to address this. So that's something to take away for the Soil CRC, and I believe that projects have now on the way looking at these two aspects by other teams. So in terms of experimental design, what we did is we used a mixed design system. So we had two approaches. One was to do a pairwise comparison, which is pictured here on the left, where we had a regenerative farm that is matched with a conventional farm in the nearby area. And those are matched with having the same soil type, same topology, and same land use. And we went and did some sampling in one field only. So the two fields are well matched. And the idea is that any differences in the matrix that we measured and we quantified, in theory, should be attributable to the management itself. And then to address one of the outcome from the SEAM analysis, we also sampled additional regenerative farm to increase the data density for regenerative properties. The reason why we needed to do a comparison is because a lot of the questions were, how does regenerative agriculture compare to what we're already doing? And so we needed to also do some measurement to allow that comparison to be made. We also, through our work with our farmer working group and the rest of the team, we came up with some criteria for selecting our sites. So there were obvious prerequisites, do we have access to the site? Is there a paddock history? We wanted to make sure that we worked with commercial farms, not just your hobby farmers, but farms that are actually producing something on a commercial scale. How to select the regenerative system, I will talk about it in just a minute, so I'll leave that one. Conventional system, so the conventional farm were selected to represent the best practice for the region, because each region has got a different set of best practice. And then I've just mentioned a little bit for the pairwise comparison, there were strict sites for matching climate, soil type, and production system. For the regenerative system, we had basically 6 criteria. So first criteria was the time since adoption of regenerative management. And so it had to be at least 3 years, as well as an understanding which was provided by the community of that is linked to the partners with whom we're working on the ground. An understanding that the regenerative practitioners in the sites we were sampling were managed by regenerative practitioners that had considered a shift in the mindset and the way they are looking at their system. And then we selected sites that had stopped using synthetic fertilizer for at least 3 years, had stopped using herbicide and pesticides for at least 3 years, had made conscious deliberate choices to increase diversity within the crop and of course the pasture system, had the livestock integrated as part of the production system, of course for the pasture it's obvious, but also for the cropping system that we sampled, and had stopped tilling their system. So this was actually quite an effort to find suitable sites. And so we sampled in the 3 regions in which our Australian partners had a presence. So in Western Australia, Southern Australia, and New South Wales. We ended up with 15 sites. 12 of which were part of a pairwise comparison, and we added 3 unpaired region sites that were an independent farm, as well as an extra region field that was part of another farm. And within that, we had a imbalance between cropping and pasture production. So we had 3 pasture sites and 10 cropping sites, because of the legacy and the relationship that our partners had with the practitioners. So let me just take you through some of our results with emphasis on carbon. So first of all, carbon stocks, what's happened with carbon stocks. So here I have plotted the raw data that we obtain measuring carbon stocks over the top 30 centimeters of the soils. The first half of the chart shows the sites that were on the pasture system, and the bottom one are the sites on the cropping system. And those are plotted by farm. So you can see that a pairs of 2 farms in New South Wales, and then Southern Australia, and then towards the right of the chart, the Western Australian sites. And in pink are the carbon stocks for the farms that were managed conventionally, and in blue, the carbon stocks for the farms that were managed regeneratively. And what you can see is that the pattern is not really obvious. And there is huge variation. So each point is a different grade. If you saw in the experimental design, we actually sampled 3 different grades in the same paddock. Each point is a different grade. So there's some variability within the paddock itself. There's variability between the farms. So when we look at that a little bit more closely, and we calculate the means across all sites, we can see that the mean carbon stock across all the conventional sites were about 25 tons of carbon per hectare, or 36 if we go down to 60 centimeters, which we also did. And then we compare that with the sites that were managed under regenerative agriculture, and it's increased to 27 for the top 30 centimeters and 41 tons of carbon per hectare if we target the top 60 centimeters. So there is a trend in an increase in soil carbon stock, but with the data we have insufficient evidence to say for sure that there is a significant increase in carbon stock under regenerative management. Our p-value is 0.2. We dive a little bit more closer into these results, and now we focus only on the pairs, on the sites that were part of this pairwise comparison. So it's the same data, but plotted in a different way. So here on the left are the pasture site, on the right, the cropping sites. And we've plotted the mean and the standard error of the mean for the conventionally managed site and regeneratively managed sites on each site. And you can see that at the bottom, conventional, regenerative. And the color now corresponds to the farms themselves. So you can see the change from one farm to the other. And what you can see is that there is a trend in an increase, but the variability is really big. And so our data basically are inconclusive in terms of informing whether regenerative management increases soil carbon stocks. We looked at the same data and we thought maybe we can decipher some patterns that are above and beyond just looking at the management. So these are the same data exactly, but what we've plotted now is the calculated difference for each pairwise comparison between regenerative and conventional management. So the line here symbolizes where if there is no difference, then the difference would be 0, right? So this is the line 0. So everything at top of this line are all the sites where regenerative management increased the carbon stock. And the ones below are where regenerative management decreased the carbon stock. What you can see at first glance is that when it increases the carbon stock, the increase is much greater. The data are really affected by one specific pair, which was in New South Wales, where there's a huge increase in carbon stock under regenerative management compared to the neighboring site. And we try to, in small letters here, I've added whether the site is a cropping system, whether it's on the low rainfall and the type of soil, because we tried to decipher whether there was a pattern due to the rainfall. One hypothesis was perhaps regenerative management has a potential for increasing soil carbon stock substantially when there is no severe limitation of water. and perhaps it might depend on the soil type, but we don't have enough resolution in our data because that study wasn't designed to decipher all the different treatment combination, soil type, climate, rainfall, management, and cropping versus pasture. So it's just the start of an indication, and what it tells us is that further research should really emphasize increasing replication within each of these different combinations. Now, if we look at the carbon fraction, so what is it's not only just about the total amount of carbon, but how does that carbon shows up in the soil? So what we've done is we've used a range of methodology. We used LICO, MIR, and wet chemistry. And this is to distinguish whether the carbon is in a stable form or a more labile form. And moving on to the results, regardless of the methodology that we used, now we are looking at concentration. So this is not about the total amount in the soil, but the concentration, and it's a better indication of the soil quality and the soil, and that's then links up to soil health. And for each of these measurements, there was a significant decrease with depth. So of course, in the topsoil is richer in carbon than the soil depths at greater depths, which is not a surprise. But out of all the measurements that we took, there was a significant effect of the regenerative management on the total organic carbon and on the hot water extractable organic carbon. So in terms of concentration, our data absolutely can confirm that regenerative management increases the concentration of carbon. The hot water extractable organic carbon, I now want to show you those data because this is one of the metrics that is proposed by multiple countries as one of the key metrics to as an integrative matrix of soil health. And so the idea is that greater amount of water extractable organic carbon might be associated with greater soil health. You can see here on the left the conventional sites, and on the right the regenerative sites plotted by depth. So the first bar is for the upper soil, 0 to 10, and then 10 to 30 and 30 to 60. And what you can see is that overall for each depth, you have a greater concentration of hot water extractable organic carbon in the regenerative sites. And now having a look at how does it look like from a biological standpoint, because there was a lot of emphasis on understanding the biology. So we used also a range of methodology, and I'm going to show you data for a few of those. And we looked at enzymatic activity because this is what informs the biological cycling of resources within the soil, and it also informs what kind of organism are there, what kind of substrate are there. We characterized the nematode community because the nematode community is an indication of where the energy that comes from the plant is then distributed within the soil. And it's a well-established method, and we also did some soil food web analysis. So I'm going to show you the data for the enzymes. And so the main take-home message there is that out of the 6 enzymes that we characterized, which were listed in slides before, the only enzyme that responded to management was chitinase. So for all enzymes, there was a significant decrease with depth, which you would expect as you go lower in the soil. There is less carbon from the plants, there's less turnover of resources, lower enzymatic activity. But what was interesting is that for chitinase, there was a significant interaction between depth and management. And basically for the top layers, 0 to 10 and 10 to 30, there was no significant effect of management, and it's just at depth that we saw that there was an increase in chitinase activity. Chitinase is acting on chitin, and chitin is a strong component, a dominant component in insect and fungi, which could suggest that regenerative system are there's more abundance of insect and fungi. I'm just going to go a bit quicker. So nematode results, what we could see here, we've plotted the relative proportion of the nematode based on what they eat. So nematode eat bacteria, fungi, there are omnivores nematode that eat everything, and there's nematodes that are mostly associated with plants. And regenerative management actually shifted the composition of the community, decreased the bacterial dominance, and increased the amount of the relative proportion of nematoda associated with plants, both for the plant parasites and the plant associates, which are usually beneficial for plants. So what are the conclusions that we can draw from our project? And the conclusion are, especially with regards to soil carbon stocks, is that we totally undersampled to actually come up with really conclusive results. We've done a power analysis, and what we've done is we're assuming we have 30 tons of carbon per hectare in a system. How many sites we would need to sample with the same kind of experimental design and the same kind of variability that we observed to actually say with confidence that we can detect a 1.5 ton of carbon increase per hectare, all the way to 7.5 ton of hectare. So the greater the increase, the easier it is to detect. And you can see here on the top left corner of the table that to do that with confidence, we would need to sample just short of 3,000 sites. And we sampled 15. And so this is really the main message to the community, to really have some confidence in our data. Either we decrease the variability within the sites, or we substantially increase the number of sites that we sampled. And so for us, really thinking about what would work well in terms of funding projects, we would suggest that because there's so much variability between the different regions, that the research projects are really designed in a way that is place-based, because there is so much differences in the different characteristics, and even what we should measure might differ between the different regions. We suggest taking a mixed method approach. There are a range of social methodology that can be used to also understand what's happening from a biophysical point of view. And there's a leading study that's just emerging from the US, where they combine biophysical measurement and social methodology to understand what's the impact of cover crops on soil health. So that's the kind of stuff we could do. Greater emphasis on citizen science, which is currently trending really strongly in the EU. And of course, increasing sample size, reducing variability, and I would strongly suggest doing time series where we're following the same site over a longer time scale. And that is it for me. So here is, while we have questions, give you a chance to write down our contact emails if you want to follow up and ask us further questions. Thank you.
Thanks a lot, Gwen. A very comprehensive and complex piece of work, and no surprise, I guess, that there's quite a complex message coming out of it. It's not straightforward. More research, more sampling is part of it, and I think a message to everyone else is that this project, as it currently stands, is coming to an end, but there'll be a lot more work done in the analysis of the results, and there'll be a lot more publications coming out of this through the Soil CRC about this piece of work. We've got a couple of questions, and we encourage you now to put questions in the Q&A box. And then if you see a question there, you can upvote it, and it will jump to the top of the list. So the one at the top of the list at the moment is from Carl Barent. Hi, Gwen. Were there any time in regen effects when comparing between the sites? I think one of the criteria was to have a minimum of 3 years in regen.
Yep. So because this project was really from a Soil CRC point of view, was just really laying the foundation to what should we really pay attention, and also how feasible it is. And the ideal would be to do a time series. So where you substitute space for time, which means you sample farms that have been converted to regenerative management for a different length of time. You could do that, or you follow over decades sites as they are being converted. And to do that, we would need to have sampled many more sites. So we can plot the data based on the time since conversion, but it won't be really meaningful because we don't have any replication within each of the different times since adoption of regenerative management.
Okay, good, thank you. And the next question is from Frank Stree. Are there any of the farmers involved in trials using pyrogenic carbon or biochar in Western Australia, South Australia, New South Wales? say feed char for livestock and or nutrient enriched biochar and or biochar compost. And then the second part is, if not yet, are there plans to investigate this in the future? I might come to the second part, the second question, but let you have a go at the first question, Gwen.
Yeah, I guess we probably should, both of us should combine our answers to this. So we deliberately kind of stayed away. Some of the practitioners we're working with are experimenting with biochar and other types of pyrogenic carbon, but we stayed away from that. Because we wanted the results to be applicable to the main practices that are seen as the branded regenerative management, and also because the Soil CRC is investing some effort in this. So I'll hand it over to you, Michael, now.
Yeah, thank you. So we do have a range of other projects, especially in Program 3 of the Soil CRC. And Program 3 is new products for soil fertility and function. And some of the work we're doing there is looking at biochar in a couple of different ways. One, as an advanced material, as a carrier for biology, for rhizobia, for other microbials, to help be a carrier, a delivery agency as an alternative to peat. But also as well as a slow release fertilizer in high pH calcareous soils, and how it might be combined with nutrients, with fertilizers to enhance its nutrient availability, nutrient carrying capacity as well. So Program 3, go to our website, have a scroll through the projects in Program 3, and then some of them we've already given webinars on, some there'll be webinars and other project outputs in the future to take a look at. All right. Next one I have from Graham Sandrell. I think it's a comment, but I'll take it as a question. Hi, Gwen. For many growers, I think they would be really interested in dollars per hectare per kilogram of carbon, or the profit per hectare per kilogram of carbon.
Totally. So that is not a simple answer, a question to answer. So the one thing I would like to say is that there is a new project that is being funded by the Soil CRC that is doing some thinking about how to answer that question in a meaningful manner. We are gathering data to try and inform that other project. So the other project is designed in a greater chance of getting a meaningful result. Our situation is that we had pasture sites and cropping sites, and in the cropping site, we can measure the total biomass, and we can have a yield, and we can have how much money was made from the yield, and we're gathering those data, and then we can relate that back for that particular the area that we sampled to the amount of carbon that we measured. But for the pasture side, it's a lot more difficult because the product is animals or wool, and they don't go in only just one site, and there is a huge variability within the farm. We didn't try to assess the total amount of carbon at farm level. That's another level of investigation. So for us, with the few sites that we sampled, we are going to get some data, but those data won't be really meaningful in a way of, like, it won't be able to be conclusive. That answers your question, I think.
Yeah, thanks. And I might take a bit of a CAO opportunity to expand upon that in terms of, and the research we're doing here. I think some people in the community are looking to us, can you tell us, does regen ag work? Is it better? How does it compare? And really, the answer is never going to happen through a scientific process definitively. Through the data we collect, through the questions we ask, we can help to inform the conversations. Ultimately, it's going to come down to individual farming situations, farming businesses, a whole range of biophysical and economic and social factors as well that will be relevant to these areas. So in Program 1, we are looking at some of those economics of regenerative farming systems, as well as some of the social aspects. But again, it's not going to end the discussion. Those but help to inform the discussion, if I can answer it that way. Right, let's keep moving along. From Rajit Kangura, what strategies are regen farmers using for managing pests, diseases and weeds if they don't apply chemicals? So it's a bigger one there for you, Gwen. You might just touch upon.
Yeah, it's a really big one. It's really context specific, and it really, when I mean context specific, it's not only the context at the region level, it's really at the farm level and at the individual level. They use different strategies. in terms of the one where I can speak a bit better to is because it's closer to my expertise, is managing weed. Already, this is why we thought a co-innovation approach was really important. Regenerative practitioners do not see weeds as weeds. A weed is just a plant growing in the wrong place. So they actually see weeds as an indicator of what's going on in the system. They have developed a whole knowledge system around using weed as bioindicators of nutrient deficiency or compaction or water shortages. A lot of these theories are not actually being investigated by classic academic science. So there's a lot of opportunity to disentangle and find out new processes potentially. So the management per se is that the question of managing weed doesn't really come into place. So it's a completely different system. For example, in the cropping system, we make sure that one of the main crop being produced was the same species, but actually the regenerative system had a range of crops. So there's high diversity, better ground cover, less chance of a weed emerging. So I think I might leave it there, but please feel free to contact me again and maybe take a few more questions before the end of the session.
Very good. Thanks. Graham Sandel again. Hi, Gwen. Most findings in the past show you need long time periods to change carbon stocks, and the large drivers are soil type and rainfall. Did you use soil types and rainfall in the analysis of this data?
Yeah, so Graham, thanks for the question. So one of the figures that I showed, when we calculated the difference between the carbon stocks in the regenerative sites and the carbon stocks in the paired conventionally managed sites, I then added a little legend indicating which ones were on which soil type and which rainfall, separating that in high, mid, and low rainfall. And this was because we're absolutely aware that this will depend on the time, but not only just the time, also how much water there is, the soil type, and whether there's clay, no clay, etc. We didn't have the resources that this project wasn't designed to address all of these questions in detail. If we had wanted to do that, we would have had to restrict the site that we sample to say one single land use and perhaps comparing two soil types with the resources that we had. So it was our choice in discussion with the Soil CRC to go broad, so it could then inform the design and how the funding should be invested in further subsequent research. So we are looking, we have the rainfall data, we have the soil type data, we don't have the resolution in our data set to test that.
Thank you. And what I took from that data too, the biggest difference between the sites, the biggest differences was between the sites, not just between regen and conventional, but between the sites which had different rainfalls and different soil types. South Australia, Western Australia, New South Wales, as you'd expect.
Yep.
I'll go to a question from Sue Briggs. Did you conduct baseline soil testing at the start of the program? If so, what was the pH and macronutrient soil levels? And probably a bit too much of an ask to give all those numbers again. That's another webinar.
Yeah, it is. Yeah, we did. We measured pH. We measured a whole bunch of minerals, in the soil. Yes, it will be another webinar. So under discussion with Michael and the team, we know that the interest is in soil carbon. So I only presented the soil carbon data. It really varies depending on the sites, and this will be in the paper. So, and please feel free to contact me if you want to follow that up later. Okay,
we'll just finish off quickly. So leave the questions that we have there. If we have other questions, please contact Gwen directly. But let's just go for the questions that are on the list. There's 3 more questions here. Hi, Gwen. What if it's from Cecile Godfrey, who's in Charles Sturt Uni leading one of our Program 1 projects? Hi, Gwen. What was the cost of data collection and analysis at each site? How can other Soil CRC projects build upon your work?
Really excellent question, Sochelle. I couldn't come up with an accurate figure right now because I haven't calculated that, but I think that's something that we should do at the end of the project and then discuss with Michael and the team. It's something that I've been looking into for my New Zealand-based work. And this is the reason why, and I think we had a conversation together, Sohail, you and I. This is the reason why I'm strongly encouraging our academic community to consider this mixed method research using a system of survey and interview to gather biophysical data, which is not our default options. It's starting to be rolled out in other countries with success. It has certain quality and it can't be done singly that way. You also need to go out and do some sampling. And the citizen approach science is also a way of lowering the cost, also in terms of the theory of change and making sure that the adoption of the results of the research is faster. That's a way of doing that. But yeah, it's quite a, I think that this kind of project really put the emphasis on how we could do research in a much more cost-effective manner, and it would be great to calculate those figure numbers so funders like the Soil CRC can then be better informed. So thank you for your question.
Thank you. These last two questions, I'll go to Jill Coglin's next. Did the project only consider cover crops, but also livestock, especially cattle, that trigger a biodiversity cycle?
I'm not really sure how to understand. So in the cropping sites, we had sheep as integrated into the regenerative management. So that was one of the criteria, is that as part of triggering that biodiversity cycle, to reuse your words, the regenerative management was using animals. Our pasture sites were mostly sheep, I believe. We deliberately didn't really, we emphasize more the selection of sites that had a dominance with sheep grazing, because we are aware that some other research efforts have been invested in looking at what happens when you graze cattle. And at the moment, there's such a paucity of data on regenerative system that we try to fill the niche and the gaps that were still left. So in a way, yes. Did we do that to disentangle cover crop versus livestock? No, because our project wasn't designed to test the specific impact of one practice. It's more the total management of the system. And yeah.
All right. Thanks, Gwen. And the final question from Kylie, but also from a few others: is there a paper we can read on this yet?
I think the paper is being written up right now. For anybody who's in academia, you will know that from writing up to publication is usually a few months. So let's just cross fingers that it goes as fast as possible. I'm anticipating that there will be some written report in a way of, diffusing that information before the academic paper is published. But the answer for now is no. Hopefully soon.
Yeah. We'll leave the questions there, respecting we have gone a few minutes over time, but recognise we're interested in this area. And just on that last one, yes, through the Soil CRC, we'll be making the results of this project available both in an academic sense, but also in a more publicly or end user digestible sense, if you follow that. But this data is fresh off the fresh from the biometrician, so to speak. It's only just recently analysed. The story's being put together. Still a bit more investigation, interpretation to be done of the results and all the other data that we've got there. But yeah, the purpose of a Soil CRC is to inform farmer practice in a whole range of different ways. Gwen, thank you very much for your presentation today. Thank you to the project team behind you, all those listed and others who've made a contribution to this piece of work. Thank you to all the attendees on today's webinar, and I just hope everyone enjoys their holidays, and I wish you well for the coming weeks. Merry Christmas. Thank you. Bye.
Thank you.